Hello everyone! Today, we're diving into a powerful technique that scientists use every day in labs around the world. We are going to explore fast protein liquid chromatography, often shortened to FPLC. It might sound complicated, but at its core, it's a highly efficient method for sorting and purifying proteins from a complex mixture. First, let's quickly understand the basic principle of chromatography. Imagine you draw a line with a black marker on a coffee filter and then dip the edge of the filter in water. You'll see the black ink separate into different colors like blue, red, and yellow as the water moves up the paper. That is chromatography in its simplest form. It's a technique used to separate the different parts of a mixture. So, what makes FPLC a special type of chromatography? The name itself gives us clues. The liquid part means we use a liquid, called a buffer, to carry our sample through the system. The protein part tells us that this technique is optimized for handling delicate biological molecules like proteins without damaging them. And fast means it's much quicker than older, gravity-based methods because it uses precise pumps to maintain a consistent, medium-pressure flow. An FPLC system is made up of a few key components. You have pumps that move the buffer, an injector to introduce your sample, a column where the separation happens, a detector to see the results, and a fraction collector to gather your purified proteins. The absolute heart of the system is the column. This tube is packed with tiny beads, often called a resin or stationary phase. This is where the separation magic occurs. When your protein mixture is pumped into the column, different proteins interact with these beads in different ways. This interaction dictates how they are separated. There are several popular FPLC methods. One is size exclusion chromatography, which sorts proteins by size. Think of it like a filter. Large proteins can't enter the tiny pores in the beads, so they flow around them and exit the column first. Smaller proteins take a longer path by going in and out of the pores, so they come out last. Another common method is ion exchange chromatography, which separates proteins based on their electrical charge. The beads in the column can be either positively or negatively charged. A protein with the opposite charge will stick to the beads, while others will flow right through. Finally, there's affinity chromatography, which is incredibly specific. The beads are coated with a molecule that acts like a lock, and it will only bind to one specific target protein, which acts as the key. As the separated proteins leave the column, they pass through a detector that measures them, often using UV light. This creates a graph called a chromatogram, which shows a series of peaks. Each peak represents a different, separated protein. A fraction collector then automatically gathers the liquid corresponding to each peak into separate tubes. This leaves the scientists with pure samples of their desired proteins, ready for further research, like developing new medicines or understanding diseases. Thank you so much for watching. If you found this explanation of FPLC helpful, please show your support by hitting the like button, subscribing to the channel, and ringing the notification bell so you're the first to know when we post a new video. Your support is what makes these videos possible. See you in the next one.